So we had just seen how David Pribnow identified that there's a consensus sequence or a similar sequence before the transcriptional start site in genes. And this wasn't only true in bacteria, this was also true in eukaryotes. And so our next question is, what is it about the sequence that actually starts transcription? Or how does a promoter start transcription, and how is it recognized by RNA polymerase? And so promoters that they notice from the sequence are in both bacteria and eukaryotes have lots of A's and T's. And so the real question was, is it just because A's and T's have only 2 hydrogen bonds as compared to G's and C's, which have 3 hydrogen bonds? And it is this weaker bonds that makes it so that RNA polymerase can melt or open up, separate those 2 DNA strands for each other? Or is it also the sequence itself? So to do that, they took a look at this. It's actually a promoter sequence from a chicken gene, and just like we saw in the bacteria, it's in a little bit different place upstream, but it is full of A's and T's, and this is what we call the TATA box in eukaryotes. And so is it the sequence that is important, or is it just having A's and T's there? And to ask this question, the researchers basically took the TATA part of it and made clever mutations. So to start, like we always want to start with our baseline or a control, is it took this TATA box sequence and, again, in a test tube, basically mixed the TATA box sequence with RNA polymerase and then saw how much transcription came out. And transcription is measured here on this blot by how dark of a band it is. So if we just focus on these lanes 3 and 4 here, then you can start to see that there is a lot of transcription when we actually have RNA polymerase added. So in addition to our positive control of adding the wild type sequence, in every other lane also we have RNA polymerase is either not added or is added, indicated here by a minus and plus, because you want to make sure that that transcription is really dependent on the enzyme being there and not something else in the reaction. And so we got a lot of transcription, so it's this big, dark, black honkin' spot when you have the wild type promoter there. But then made 2 different mutations. And one of them changed the second T to a G, and then in the other experimental condition changed that second T to an A. And this was really clever because, again, it's asking, does it matter that it's a T or does it matter that it's just 2 hydrogen bonds there? And so and then if we look at the data here, you can see that the results are that either the TAGA, when RNA polymerase II is added, has a small amount of transcription, or the TAAA has a small amount of transcription when RNA polymerase is added. So that tells us that it's the sequence is important and that it's not just A's and T's present.